Welcome to the Cutting Edge, where we explore the latest scientific breakthroughs that shape our understanding of health and disease. Today, we delve into a fascinating study called Cognitive Decline in Sprague-Dawley Rats Induced by Neuroplasticity Changes After Occlusal Support Loss, conducted by Xiaoyu Wang and colleagues from Capital Medical University and Beijing Stomatological Hospital. This research investigates how loss of occlusal support, tooth loss, affects cognitive functions through changes in neuroplasticity. Background on tooth loss and cognitive function. Tooth loss is a common issue, especially in older adults, and has been linked to cognitive impairment. The most well-known cognitive function impacted by tooth loss is memory, particularly those functions associated with the hippocampus. The hippocampus plays a crucial role in learning and memory, and its function is heavily reliant on neuroplasticity, which involves the brain's ability to reorganize itself by forming new neural connections. Despite various proposed mechanisms such as reduced metastasization, enhanced neurodegeneration, and chronic inflammation, the exact pathway through which tooth loss leads to cognitive dysfunction remains unclear. The study aims to shed new light on this by exploring changes in neuroplasticity and related synaptic factors. Study objectives and methods. The primary objective of this study was to investigate the effects of occlusal support loss on spatial cognition and neuroplasticity in Sprague-Dawley rats. The researchers focused on the following key areas: the impact of tooth loss on spatial cognition, assessed using the Morris Water Maze, MWM, changes in the expression of brain-derived neurotrophic factors, BDNF, and synaptic proteins such as PSD95 and NMDAR in the hippocampus. Structural changes in the ascending nerve pathways observed through Golgi staining and immunofluorescence IF. Methodological approach. Animal models. The study used 36 male Sprague-Dawley rats, divided into young, 5 weeks old, and middle-aged, 8 months old groups. Each age group was further divided into tooth loss, experimental, and control groups. Tooth extraction. Bilateral maxillary molars were extracted in the experimental groups to simulate occlusal support loss. Behavioral testing. Spatial cognition was evaluated using the Morris Water Maze. Molecular analysis. Quantitative real-time PCR, qPCR, and Western blotting, WB, were employed to measure the expression levels of BDNF, AKT, PSD95, and NMDAR. Historical examination. Golgi staining and immunofluorescence were used to observe structural changes in the neural pathways and the hippocampus. Key findings. Cognitive decline. The Morris Water Maze test showed significant cognitive decline in both young and middle-aged rats following tooth loss. Rats in the tooth loss groups took longer to find the platform and had fewer platform crossings compared to control groups. Gene expression changes. qPCR results indicated significant decrease in the expression of BDNF, AKT1, and NMDAR2B in the hippocampus of rats in the tooth loss groups. The expression levels of these genes were significantly lower in both adults and middle-aged rats with tooth loss compared to their respective control groups. Protein expression changes. Western blot analysis revealed decreased levels of BDNF, AKT, PSD95, NMDAR proteins in the hippocampus following tooth loss. These decreases were more pronounced in the young tooth loss group. Structural changes. Golgi staining showed a reduction in the number of neurons and nerve fibers in the ascending sensory pathways from the periodontal tissue to the hippocampus. Immunofluorescence confirmed downregulation of BDNF and AKT in the hippocampal CA1 region, indicating impaired synaptic plasticity. The study provides compelling evidence that tooth loss induces cognitive decline through negative changes in neuroplasticity. The reduction in masticatory activity following tooth loss leads to decreased activation of the neural pathways connecting the periodontal tissue to the hippocampus. This in turn results in lower levels of BDNF and synaptic proteins, which are crucial for maintaining synaptic plasticity and cognitive functions. The findings highlight the importance of maintaining oral health and preserving occlusal support to protect cognitive functions, especially in aging populations. Further research is needed to explore the potential for dental interventions, such as prosthetics or implants, to mitigate cognitive decline associated with tooth loss. This study underscores the intricate relationships between oral health and brain function. By elucidating the mechanisms through which tooth loss affects neuroplasticity and cognitive function, it opens up a new avenue for preventing and treating cognitive decline through dental care. Thank you for joining us on this episode of The Cutting Edge. Stay tuned for more insights to the latest scientific discoveries that shape our understanding of health and medicine.